we are ready to play with RuleBender. So we got RuleBender up and going, and there are a few. This is RuleBender is based on the Eclipse platform, and which is meant to be a programming environment, a so-called IDE for Java and many other languages. It has a very rich interface with lots of features, only some of which we're going to explore today. There's a little bit, because it's a special purpose tool in a way, it has a certain learning curve. There are certain ways of doing things in Eclipse that you have to learn a little bit about to be able to effectively use RuleBender. It's not unlike CellBlender in that you've got Blender there, except I think it's a little less intimidating than Blender. But that's up to you to decide. Anyway, so what you're confronted with now is like a completely blank set of things. This is what you get when you install RuleBender on your machine. So the first thing that we can do is, so everything in I'm going to keep saying the wrong, I want to say Blender, but everything in Eclipse is based around projects. So the first thing we need to do is to create a new project. We've created a shortcut here for creating BioNetGen projects, so you can just click on File, New BioNetGen Project. It gives you a little warning that it's going to ask you to do something. So basically, we can select a default sample file to go into the project. So let's just pick, for the heck of it, template.bngl, and we can name the project something. So let's just call it, I'm going to call it LV, because we're going to do a simple Latka Volterra as a warm-up example. So so the template model comes up as template.bngl. We can, if you should be able to click on it. I was going to try to, if you do a copy and then a paste again, you can make a copy, and I can just call this LV BNGL. So it doesn't matter what you call this file, but whatever I just made a copy of template, so we keep template in a unchanged state, and now we can add the components that we need to the model. So the BioNetGen composed of a set of blocks. There are, I think, 6 block types, and we don't need all of the blocks for every model, but it's useful to know what they all are, and we'll go through some of those. So let's just make we can make our reaction rules first for the Latka Volterra model, and as we did before yesterday. So we had S and I think he called it predator and prey. I call them sheeps and wolves, so S and W. So for example, the first rule of the Latka Volterra model could be S, and I'm going to put parentheses next to the species name to indicate that it's an object. So we have S goes to S plus S. That's our first reaction. And does anyone remember for the reaction limited case what the rate constant is? I think it's 1.3E5. 1.29. Here it is, yeah, 1.2. Here's okay, good. I have a cheat sheet. This is good. This is what I want. So this is the model, but don't look. Is that better? Okay. So we have our rule. It's 1.29 E5. So I should mention that. So we could use a parameter here. We could call this. We could give this a name and set the parameters up in the parameters block. But sometimes it's convenient just to have things in place. So we'll do that for now. And the parameters in BioNetGen model are, there's no set of units that's required. You just have to define a self-consistent set of units so that you pick some concentration units for molecules, species concentrations, basically. Unimolecular rate constants will have units of 1 over time. Bimolecular rate constants will have units of 1 over concentration, 1 over time, and so forth. So we'll see that we can use that to our advantage. When we want to make a model that's compatible with M-cell, then we become more restricted in our choices. But OK, we won't worry about that for now. So we have S goes to S plus S. We have sheep get eaten by the wolves. And for that one, the constant is 1 was 10 to the 8th for the reaction limited case, divided by Na times the volume of the system. So here, the 10 to the 8 was 10 to the 8 per molar per second. And in order to convert that into molecule count units, which is what we're going to use since we want to have discrete sheep and discrete wolves, we need to divide by Avogadro's number times the volume of the system. And we'll put the volume in. So we need to define the parameter for the volume of the system. So we could actually just go up to the parameters block and do that now. So the volume. So it's useful to remember that a cubic micron, which is the unit of volume in M cell, is a femtoliter. It's 10 to the minus 15 liters. So if we have volume, if we put a factor of 10 to the minus 15 up front, then we can put in the volume that we had for our M cell volume for this Latka-Volterra, because we want to compare this Latka-Volterra to the one in M cell. Yep.
Does indentation mean anything?
I'm sorry, I missed what you.
Does the indentation mean anything, or is it just an A?
It's just for layout. Yeah. The white space is the white space at the beginning doesn't matter. You need white space between the tokens. Within a token, within a reaction or a complex or a molecule, you can't use white space, which can be a little bit annoying, but there are reasons for that.
So the end of a rule, if it is longer than one line, I mean,
you can continue with a backslash. I'll show you an example of that. You can continue lines with the backslash. So I could put, for example, like this. That's valid. If I do this, though, you'll get an error. Yeah. If I save that, you'll see there should be an error there. Right. Oh, it's going to crash. Beautiful. Nope. Oh, it just gave an error. Some problem processing. Yeah. And oh, it was actually this. It found the syntax error up here because the expression is incomplete. Anyone remember what the dimensions of the box were from yesterday? I think it's 0.0. It's 0.5 by 0.5 by 0.01. So you can see that we can do arithmetic in expressions, just like in MCell, in CellBlender. Right. So now we've defined our Avogadro's number, and we defined the volume of the reactor, and the system is, when you save this, the syntax is automatically checked, and the fact that there are no errors meant the system was happy. It's a good idea to define molecule types, just like it's a good idea to define your variable names in a program. So here we're going to have unstructured sheep and wolves. You'll notice that if I start to define molecule types in this molecule types block, so I defined S, but I didn't define W. So as soon as I define one type, then that turns on type checking, and all types are going to be checked in other blocks. So when I set the molecule type, when I defined S, now I'm getting an error because W isn't defined down here at line 22, where I have the predator-prey reaction. So I need to define W up here also if I'm going to define. So it's kind of an all or none thing with types.
Why are we normalizing the rate constant for S plus W with rate number?
It's a bimolecular rate constant, so it has units of 1 over concentration, 1 over time. So in order to put it into count units, which is what I'm going to use for the sheep and wolves, I want to have them be discrete. I need to divide. For the first one, it's unimolecular. So unimolecular rate constants have just units of 1 over. Great question. Thank you for asking, because yeah.
I'm not getting any picture. Should I?
When you save, you should get a picture. Yeah.
Oh, got it.
The picture, unfortunately, is not as informative as one might like for this model, because the contact map only displays one agent, one object of each type. So something like S goes to S plus S it has a kind of funny representation. So if I comment this out and I look at just that reaction, so this is just the rule, the multiplication rule. So you see that S goes, this is going out to a node, this represents a reaction, and then it's creating an S, but you can't see the stoichiometry. You should be able to right click on the, I had trouble with this too. If you right click on the object, you can see R1. Yeah, it's not really meant to visualize this kind of rule. But, and so you can see what that one looks like. You could see that this is what the single predator-prey rule looks like. So you see sheep going in and wolves going in and only wolves coming out.
Yeah. Are you saving in between each?
Yeah, you have to save to update the map. So the contact map is only created upon or updated upon saving.
But I'm seeing wolves coming out and not going in. Me too.
For this one? If you see both, so if I have both up, it looks, you get it randomizes the layout a bit. So actually, once the model is set, the BioNetGen, the RuleBender will preserve the layout. If you move things around, it'll come up the same way after you've, I can close this, for example. I can reopen it, and it'll come up. Oh, I didn't save it. Oh, that was beautiful. Wow. Oh, no. What happened? Oh, it's because I edited. Oh, I see what I did.
That was the wrong one.
I edited the template instead. All right. Yeah, I guess it's easy to do that. So as long as we have one call, it doesn't matter. That was just for fun.
Sorry, can you explain the first arrow again? Like, why does it have S going two ways into R1?
This one?
Yeah, why is that an exchange, not a...
So S is consumed by the first one, but two S are produced. It doesn't show the stoichiometry of the S being produced, but there's two, which I can. This visualization just doesn't capture that. It's really meant to show. This is really meant to be a contact map that shows molecules, their binding interactions, and what states get modified. It's not so good at showing production and consumption of whole molecules. So it just wasn't really designed for that. This one is useful. I mean, you could see for this one that there's sheep and wolves going in and only wolves coming out. So you get the idea that there was something happened there. Okay, and then if we add wolves going to, so here instead of NULL we could just do 0. We just call it 0, the null state. And if we make that 1, 3, E5, so now we see there's a node where there's just wolves going in and nothing coming out. So this diagram I think is actually pretty descriptive of the system. Okay, so there are two things left to do here. One is we need to define the initial state of the system. So we need to basically put molecules into the system, just like we did with molecule placement in MCell. The seed species are the molecule placement in BioNetGen. So we need to put some sheep into the system, and I think it's 1,000 and 500. So if we put 1,000 sheep and 500 wolves in, it looks like that. So everybody got that. And it's useful. So just like we defined our molecule counts, our plot, what do they call plot outputs in CellBlender, we need to define if we want the default plot that comes up when we run the simulation to show us the outputs of the model. We need to define those. So we can. I define two variables: S tote, which is the total number of sheep, and W tote, which is the total number of wolves. So this just says, match all the S molecules in the system. Here, there's no structure to any molecule, but and here are all the wolves, and that's it. Functions we don't need for this. We could just delete that block. Oh, I forgot something. So each of the observables has a type. We need to call these molecules. This is a molecules observable. It means count every instance. There's another type of observable in which we count as one instance every instance of a species that matches a particular pattern. So if you had a complex of sheep, for example, and you had a molecules observable would count 2 sheep for a complex of 2 sheep. A species observable would just count 1. It would count 1 instance of a complex containing 2 sheep. We'll come back to that in another example. So now we have everything we should need. Now I just want to check the so the integration time is very, very short by BioNetGen timescales. It needs to be 0.001, so 1 millisecond, and I think I did 1,000 time steps. And semicolons are not required at the end of a line in BioNetGen. That's kind of Perl syntax, but we don't need it. And that should be it. And so now we should be ready to run this simulation. So to do that, we go over to the simulation tab here, and we click on that.
Sorry, where are you?
On the right side of the interface. Top right up here. So the contact map was where we showed the diagram here. If we go up and click on this simulate button here, we'll see this dialog, and we should see that the file I'm editing is template.bngl. That's selected here. If it weren't, I can change the selection by clicking on the appropriate item. This, remember, though, is a blank file, so we want to be clicked on template.bngl. Run file. There are 2 options here. There's run the simulation as specified in the file. There's also a parameter scan that we'll get to. So we hit run file. If we hit run, we'll see some stuff happening. The log file is written here, and voila, we have predator-prey dynamics.
Huh. Yeah. I don't know.
How many people didn't get that?
Yours converted.
So is this
Wow. Can you go back to the?
Because we used a different. Look, our time is different.
Oh, did you guys set? Did you set the time, the tn variable? I changed the parameters of simulate ODE, and if you didn't do that, you may see something quite different. It should be 0.001 and 1,000 steps.
I mean, it shouldn't matter, but
what environment is this? Or is this some specific
RuleBender is its own standalone app, but it's built in Eclipse Rich Client Platform.
Okay. I guess what language is being written here? Is this something specific?
Oh, what language is the BioNetGen in?
Yeah.
It's its own thing, but I mean, it's being processed by Perl. So it's syntax that's sort of based on Perl, but the blocks are, just.
Because it feels very much like Eclipse runners. I'm wondering if we could, open up some other IDE and run this same program.
You could pretty easily within Eclipse. Yeah, you could. Yes, you could definitely. People have defined highlighting modes for other editors, but I don't have any current ones. The parsing, the syntax highlighting here is being done by a parser that actually understands pretty much the full syntax of the language. There's some complexity, like to realize that a token like W is not defined in molecule types is more.
Where did you get
The
the plot?
plot is the other window that's up here, the tab that's next to the. Yeah, sorry, I forgot to tell you how to save. It's just command. It's just Control S, right? Of course, or file save. And if so,
When you run, there's something that says run file, and
Yep.
then there's something that says run. What's that about?
Run file is that. So you see that there's the selector over here. There's two types of running. There's run file and parameter scan.
So it's just the type of.
Yeah, and then run is the actual action button that does something.
And what's the species graph?
The species graph will show you, let's see. So if we go here, it will show you the individual species. Here it's not going to be very exciting because there's no real complexes. But if you click on, so if you go to the outputs underneath the plot, each of these molecules observables was defined as a, it's selecting a set of species in the model that match it. So you can see here if you click these items, it will show you a graph of the complexes. It's really, really not exciting here. But when we have complex species, it could be useful for debugging because you can see all the different species that are in the model. Okay. So everybody got this. I think that was all I wanted to do with Laika Volterra, because we can't. So you could compare this output for the same parameters with your CellBlender simulation, and see if you know how similar. The results should match pretty well because this is parameters. These are in reaction limited regime, so systems should be effectively well mixed, and you should get oscillations with similar frequency. Or they should match pretty closely, if not exact. Oh, I can show you one other thing. So there is stochasticity. So we simulated this with ODE. Oh, and the template model is bad. So we used the ODE method. The proper syntax, actually, the preferred syntax, is to use a method flag here, and there's two options for the method variable. The method parameter for simulate method. It's ODE was the one we just did, but if we switch this to SSA, now so that was why we set the model up with discrete particle counts. So now we can just switch to SSA and do a simulation of the same model, but stochastically. And we can see now that we get very similar, oscillations have very similar period. Sorry about that. So here, so you have to, and BioNetGen will carp at you, or RuleBender will carp at you if you don't put double quotes around the SSA. So you have to enter the syntax just as I specified here.
And it doesn't matter if you have a space after the comma or not.
No, the spaces in the argument list are robust there. But it's in the complexes. It's in these guys. Tokens that you can't put spaces. So if I put a space here,
Yeah.
it's an error.
And if you don't put spaces between the pluses, it reverses to that.
Yeah, no, that's fine.
Okay, yeah. So
I hate that, but that's how it is. It's it turns out to be because of it's this space that causes that problem. That if we had a separator for the rate constants, then we wouldn't have that space, that white space problem. And it's one of those things you figure it out, but it was too late, and we had the syntax, so it's hard to roll back now because it would invalidate every model previous to. So anyway, that's just an aside. Yeah.
Sorry, my system is expecting some R bracket. I mean, I followed the syntax that you added methods in, but somehow the system says that there is a syntax error there.
I mean, you're still saying simulator needs.
Yeah. You need to get rid of the ODE up at the front. That's the template file that it came with is actually dated. I'm sorry? You could, yes, simulate but that's deprecated syntax. Just it's still back. It will work. Yeah. Because you can also change this to NF. And watch, yours won't work, but mine will. And it will do exactly the same simulation, but using an NFSim instead of Gillespie. And it should give you more or less.
Different heights in the peaks.
Sorry?
Now we have different heights in the peaks, like.
Yeah, because they're completely different methods to do the same simulation. So there's no possible way you could get the same sequence of event generation. Well, I shouldn't say no possible way. It would be very tricky to accomplish that objective.
So if we run this multiple times, will we have different seeds and get different results?
Yes. It's not M-cell.
So how do you get,
The same seed again.
You can say there's a seed option. You can say seed like that. I didn't say it aloud because I want to make sure it works. And here's really fancy Eclipse stuff. I split it by. Ooh, I have to make sure I have.
There we go, okay. And then you can put it back in again.
That's the wrong one.
If we drag this window out, you can put it somewhere else.
Damn, I don't think that worked. I don't. Here, hang on one
I
second.
think it did work, it looked like a different result.
Well, right. That's what I'm saying. If you want to make the same, so if you NFSim may not be recognizing that seed. But I'm just doing it with SSA once. There's one pattern, and then I'm doing it with SSA again.
And then do the different things are the same.
Now it's the same
Yeah.
because I specified the seed. But NFSim didn't take that seed variable. That's a flaw.
I think it did.
You think it was the same?
Now it looked different, and it should have looked different because you didn't have a seed the first time you ran.
Oh, I see your point. All right. I'm modeling good practice. Every question you have, you should make sure you know what the right answer is before you move on. Okay. Thank you, Tom. Okay. So you can set the seed, but by my default assumption is I want to get a different answer every time I run. Like I don't want I want it so that if I forget to set the seed, I get just different stuff. Now, in the log file, it will tell you, BioNetGen will tell you what seed it used. So you could reproduce the simulation if you wanted to by setting the seed.
It uses a random seed.
It uses the
random
clock.
random seed.
It uses a system clock to seed the random number generator.
And
It looks pretty good now, though.
in MSAL, we just made a different policy
Yeah.
decision that if you don't say what seed, it's always seed 1.
Right. But see, the reason that I designed it that way, I know I always generally want it to be random, but I'm often going to forget to set the seed. And so I really want to avoid the situation where I run 100 simulations and then I do it again and I get the exact same thing and I go, damn, and I just wasted an hour of. So yeah, it's just a question of which default you prefer. All right, so I don't think I wanted to show anything else with Laika Volterra.
How do we?
But if you want to see something else with Laika Volterra, tell me, or we'll move on to the next thing.
Wait.
So Joel and I made that policy decision about the seed in some of the earliest workshops that we held in MSAL, where we had users who were confused that that it was different every time. They wanted it to be the same every time. So we had the opposite input from our users.
It's kind of reminding me, like if I take a coin out of my pocket and flip it, I want to get the same answer every time. Otherwise, or I randomize the coin first. I don't know. It seems like.
I would speak for trying to train students in this that you might like to have a whole class of people who are getting the same result for starters.
That's exactly what the students were confused.
I want them to be confused
Well,
at first.
they're not willing to ask questions. They're just confused with no purpose.
Well, then tell them that they're going to be really confused if they go in.
Because you all got a different answer than I did, I'm sure, right? Because the seeds are different on these machines. There's no way I could make I think to make it the same between your platform and mine is pretty difficult to do.
Unless you knew what seed it was.
Yeah. Well, I'm not even sure.
In Excel, we have a built-in.
You have it built. I don't. We use the
That gives you
system.
the exact same stream of numbers on
Yeah.
every platform available.
No,
on
we don't do that.
exact same stream of numbers.
So it's the same randomly generated.
Yeah. For C1 means a certain stream of numbers.
I'm going. So, all right.
It's really hard to achieve.
So what I want to show you actually before we go on to the next model, let me just show you so if you go to bionetgen.org on your web browser, I guess I have to get wireless. What's the access code? N.
NTW
Uh-huh.
45 B 9E.
Okay. I accept the terms of use. No, that wasn't helpful.
You have to collect it. Oh.
I did. I can do that too, yeah.
Oh, it might be case sensitive.
Yeah, I think they
It
will
was all
have
caps.
those.
All caps.
It wasn't yesterday, but
Yes, it was. I mean,
it was.
Yeah.
So,
All caps,
Capital N.
NTW. In all caps, NTW, yeah, but all caps on.
Capital B.
Capital B, yeah.
I accept the term. Okay, we agree that it's exactly right. Success. Okay. All right.
You can just type it in something.
No, it's not. Okay, bionetgen.org. When you want to get the software, you go to download. For the purposes of what we're going to do today, just the tutorials. And there's a whole bunch of models here that demonstrate many of the features of the language. The Latka Volterra is actually the third example. This is what we more or less just did. Here I used parameters K1, K2, and K3 and defined them up in the parameters block. I hope you guys, can you see that at the back more or less? I can make it one bigger. But otherwise, the model was pretty much the same as what we just did. It's very easy to copy and paste the text of these models out into a blank file in RuleBender and then just run them. And I set that up because if you make certain typos in BioNetGen can cause a lot of mischief, and so just to make things go faster when we wanted to do tutorials, I set that up. So if you go to lr.bngl, this is just a simple ligand receptor binding model. So why don't we just actually download, just practice this copy and paste method. We just copy that text file. In RuleBender, we can go make a new file. We could call it lr.bngl.
Which one are we printing down? LR.BNGL?
Yeah. Paste. And when you paste it in, it comes in, it formats it for you. And when you save it, you should see a nice contact map showing a ligand binding to a receptor. Raise your hand. If you encounter a problem, just raise your hand and we'll try to
How do
help.
you make a new file? New BioNet?
You can either go, you can do new here and do a new file using that wizard, or what I just do is I right click on the directory, new, and then file. And it'll bring you right up into,
The wizard results.
yeah. The wizards are, they take a little getting used to. Once you're used to it, it's like, it's fine, but it's not like completely standard. It's something new, so.
What's Toy Jim?
It's another example model. It's a very old model made that was, yeah. So yeah, there's a bunch of them on there, and we'll play with some more, time permitting. But this is a nice one because it just shows it's the simplest binding interaction. We already saw an example of a rule kind of like this before. But now this is the first model that we have where we have structured objects. So we see that the molecule types, the ligand molecule has a binding site for R. This is a standard convention. If you're modeling sort of generic protein molecules that have cognate binding sites for other protein molecules, and we don't have a name for that binding site, we typically give them a name that's the cognate of the protein name. So here, L has a little r binding site for the big R molecule, and vice versa. And so the standard default for the initial molecule placement would be to just place unbound molecules in their, and if any of the components have a default state, just set the default state to be the whatever its default value is, typically like unphosphorylated, for example. And then the initial counts are given by L0 and R0. And here L0 we converted from a concentration in molar to a copy number. So it's a good idea to make sure that that number actually comes out to be something reasonable before we, depending on what kind of simulation we're going to do. What did I do wrong? Lig const is 1e minus 9 times, there we go. So we see that I was just using the calculator to figure out that it's going to give me 602,000 ligand molecules here that are going to be, that's what L0 will come out to be. So for the observables now, they're a little bit more interesting than what we had before. We have two observables. One is the number of free receptors. So this is the receptor molecules that have no L, that have no ligand bound, L bound to the cognate site of R. So we represent that with the pattern RL. And then the bound molecules, this is one way that we could represent a bound molecule. This a receptor that's bound to a ligand. So here we have explicitly the ligand molecule, its receptor site is bound to the L site of an R molecule. There are, in BioNetGen, we have wildcards to represent binding states. So we could make another, we could just call this bound to. There's another way to represent the same structure using a wildcard where we just convert this bond to a plus. That plus is a wildcard that says there has to be one or more bonds to that state L, to that site L. So we can see that when we plot, these observables should be identical.
And then we can make another reaction and see that then if we say that the complex combined enough of the ligand.
Right. If there was another, right. Yeah. So if we had another type of ligand, then this would match that as well. So if we had L2 and we bound it to the same L site, then this one would be different now. So that's the danger also. James makes a really good point. If you use a wildcard like this, the meaning of that is going to be model dependent. If in this particular model where there's only one type of ligand, it means one thing. If I add another type of ligand, now this observable means something different. And we have to be aware of that. So with convenience, there comes a cost. Right. Okay. So you run this, and I'm guessing that this is a very, yeah, all right, this is about right. So we see that the system rapidly equilibrates, and it's kind of boring. It reaches a steady state pretty quickly. I can zoom in by selecting the area of the plot that I want to zoom into. So the free R on a 10-second time scale binds to the ligand, and we reach a steady state in which exactly half of the receptor molecules are bound. How do you unzoom? Select backwards. You can also right-click and zoom in, zoom out
With the middle mouse though.
Okay, so maybe it's just the fact that I'm not using the right mouse.
On a Mac, it's the left but it's the regular left button. On the Linux machine, I have no idea.
Left click a certain area and then
Yeah.
Select it to zoom in.
Yeah,
Yeah,
that's what I was.
trying and it wasn't doing it.
But yours is using the middle.
Yeah.
I think you're right. If you're too close to the axis, it for some reason doesn't like it.
Oh, yeah. No.
Yeah, it's not perfect, the plot window, but it's pretty good. I mean, so it's very nice. You can, when you're building up a model, having this loop closed for you that you can just hit a button, get a plot, change something, and do that, iteratively is very convenient. And one thing I should mention, so every simulation that you run, so every time you hit the run button, BioNetGen is collecting the results, or RuleBender is collecting the results in this results folder, so for every project, there's going to be a results folder for that project. If you navigate into that, if you open that up, you see that you're going to have they're grouped by the model. So we just set up this LR model, and we see that for LR there's one set of simulation results. They're time stamped, so the timestamp is set up so that they'll always be in order from oldest to newest. So every simulation that you run is saved. The input file, the output files.
The Excel file, XML file. Where's
There's the XML file is something that's where did that come? That comes from a command. I don't actually know.
I'm just trying to figure out if there's
I
a
don't-
file that tabulates the graphical data so that we can regraph it.
Yeah. Thank you for reminding me to tell you about that. So two things. There's two ways. So undoubtedly, the people who do a lot of stuff on computers, you want some plotting package other than what you've got here. This is just simple, quick and dirty, but it's not great. So two things that you can do. First is you see that at the bottom of the plot window, there are two tabs, Chart, which is what is selected. If you select the other one, it gives you the data in tabular format. So this is just plain text file. You can, if you hit Control A, you can select all the text there, and you can copy and paste that into whatever you want. So you can copy and paste that into a text file. That text file already exists, so you don't really need to make another text file unless you want to. But you could copy and paste it into Excel or whatever. I won't demonstrate Excel because my Excel skills are not great. But so yeah, so you could, and I actually often just look at the data. It's a little bit cumbersome in zoomed in this far, but I often just look at the tabular data because sometimes looking at numbers is easier than looking at a plot. Like you can, like if you want to see the two time points in, between which something changed value particularly, this is a good way to look at that. So.
I don't see the chart, the button to go back and forth between the chart and data on the.
It may look slightly. Can you guys see that? It may look slightly different for you compared to how it looks for me.
For what?
For the chart to the LR jet. Okay, you got it.
Okay. Well, I don't actually see the contact map
There's
at all.
chart and plot.
You don't see the contact map at all,
Chart and point or something.
ever.
Gray
And then below that, there's
in
chart.
none of the examples, but I will say that my Java's not updated. It's like one behind or something.
You have 1.7?
I don't have 1.5, I have 1.6 something.
Oh yeah, if you have Java 1.6 on your Mac, you won't see. You have to update to. You have to get the JDK for 1.8. Although with the newest release of RuleBender, when the current 2.2, that's true. When we update that, it'll come with Java, and you won't have to install a separate Java.
Well,
But, yeah.
I got the 2.1.0.8.
Yeah, same. You need to have 1.8 for that to work, for the contact map to show up. You're also missing I think it won't give you error messages from the, if you have a syntax error in your model, it also won't show that properly.
It gave me an error message even though it ran the simulation perfectly.
Right. That could be that can happen for other reasons too. There's the parser that checks, the parser that's used by the editor to check for errors is different than the parser that's used by BioNetGen when it runs. It's generally stricter, as you'd like. Like it's rare that you'll get a BioNetGen error if you don't get an error.
It's the parser. But
there can be issues if you have if you have an old Java, then the parser may not work properly either. Yeah.
Say again which Java
1.8. There's a for the reasons. Yeah, let me not. I won't even mention it. If you have problems with it on a Mac, let me know and I will help you. I'll just leave it there. Okay. So yes, the question Alec mentioned. So we looked at these. So I was telling you about the navigator. So you can see that the results from every simulation run are kept there. The input file, the GDAT file is exactly the file that you can browse here. It's just a tab file with the time and all the observables that you defined in tabular format. When you double click on one of these two CDAT or GDAT files, you'll get a plot. So in the CDAT file is interesting. That contains, for a model where you generated the reaction network, it contains the time courses for every species in the system. So here there are 3. So if I click on that, I'll get a plot. So RuleBender recognizes that it can plot this data, and so it shows you a plot of all the species concentrations. So if you go into the outline window here, you can select which variables are shown. Sometimes it's useful to uncheck them all and look at them one by one. So like if you want to look at the bound complexes, you check box number 3. This is also in the CDAT file, you can also navigate through all the species that are in the model. So if you go to the species graph, it will display the currently selected species when you select it in the outline window. So you can see all of those guys. Again, not too surprising for this model, but sometimes it's useful to be able to see a graphical representation, of the string, because that could be very long. So, and we can select all of these. This shows the time course nicely. You can change the options here as well. You can see the time points. Sometimes that's useful. All right, so that's about it for navigating. The other files of some use to you, possibly, would be that there's the network file, which shows the actual reaction network file that the simulation is being run from, and that BioNetGen produced by expanding the reaction network into all possible species and reactions. So here, remember, we started with L and R defined, and BioNetGen discovered the LR complex, and it set its initial concentration to 0 because the LR complex wasn't present in the seed species, so it assumes that the concentration is 0 by default. It also shows you all the reactions that were generated, and to understand what this 1, 2 space 3 means, you have to realize that the numbers here refer to the indices of the species. So this first reaction, reaction 1 on line 17, says species 1 plus species 2, that's L plus R, goes to species 3, goes to the LR complex. And then you can see that the reaction on the next line is just 3 goes to 1 plus 2. So it's just the reverse of that. There is an option to get the reactions printed out in text form that's more human readable. But for simple debugging purposes, this is sometimes useful. And I think that's all I'll say about the net file. The net file format is described in gory detail in the documentation, and sometimes it's useful to look at that. All right. And the last thing is the log file. So BioNetGen will keep a record of everything that was done within the simulation, and you can see. So it's telling you everything that's happening as the file is being interpreted. This This part where it's showing the iterations shows you the process of network generation. You start with the 2 seed species and 0 reactions. The first iteration expands the set to 3 species, and there's 1 reaction. It found the forward reaction. And in the second iteration, it finds the reverse reaction. And then, since the number of species didn't change between the iterations, it stops. It's found all the species, and we have a full network. And then this shows you that it ran. It says it's running the simulation using CVODE. That's the ODE integrator. It's writing the file out. It's writing the network file out. Then run network reads the file back in, and so on and so forth. So this can be useful, again, for debugging, making sure that BioNetGen did everything you thought it was supposed to do, making sure that certain options that you used in the simulate command were properly registered. One thing we can do real quick is to go back to LR and change the method here from ODE to SSA, just for the heck of it. Since we use counts, we can legitimately do that again and run the simulation. It takes just a little longer, and if you squint, you can see that there's some noise. So this is a model where the stochasticity has a very small effect on the result because the copy numbers are high enough, several thousand, that the noise is a small feature. But you can see that the noisiness very easily when you look at the tabular data. The number of free receptor molecules is jiggling up and down. And I did this. I just wanted to show you. Okay, now we see a second entry in the results for LR, and if we look at the simulation log, we can go down and see that we can see the output for running SSA, so network simulation using SSA. It gives me the simulation ID. Where's that seed? Huh. My whole point of showing you this log file was to show you where the seed was, and now I don't see it.
Maybe underneath the simulation ID it says run network 3.0.
It does, and then reaction update.
Search for seed like control F.
Yeah, it's not here.
Maybe it doesn't tell you.
It should tell you, damn it. I guess it doesn't anymore. That's
Maybe
very.
you can try putting a seed in and see if it ends up.
Probably it does.
Does it map the order of your
It tells you the process ID, but that's not the same thing. Don't know what's up with that. Anyway, if you did, it would be very easy to hack it to make sure that the seed did get put out in the log, and it should have come out. But anyway, the other point too is that the log file is automatically constructed for you down in this bottom window under console, so you see that every time it's run as well. But there is no seed in there. All right. Good that I checked. I cannot make. I was going to tell. I told you before that the seed was there, but you see that I'm wrong. Okay. So one other thing that we can use this file to demonstrate is that we can the one other there's oftentimes with the model we want to look at how a parameter changing one of the parameters affects the output of the model, and for that we can use a parameter scan. There are two ways to run the parameter scan. One is to make a parameter scan action down here, and so instead of the simulate command we can use a parameter scan command, and I'll show you that in a sec. But the simpler way to do it is just in the simulate tab here. Instead of run file, we can select parameter scan. And then we get a dialog box with some options to fill out. So if we want to scan a particular parameter value, we can do that. Let's scan the ligconst. And we can do that from a value of 1e minus 12. Let me just cheat for 1 sec here. All right, I picked the right one. To 1e minus 6. So from picomolar to micromolar. So just to make a reasonably smooth curve, we can pick 50. Now, the trick is, I'm sure most of you know this, but if you don't, you should know that if you want to do a parameter scan over many orders of magnitude, you'd better log scale the values. Otherwise, you're not going to get very much of interest. You're just basically going to see how the parameter varies at the far end of the range. So frequently, when we don't know very much about what value to set a parameter, we scan it in log space over multiple orders of magnitude, and we see what happens. So the next flag that you can set here is this steady state option. And that has BioNetGen check to make sure that your simulation has reached steady state by the end time of the simulation that you specify. I don't usually use this because once in a while I have a model that doesn't reach steady state for various reasons. But we could check. Let's just live dangerously and check it and see what happens. But typically what I do is I just make sure that my simulation time is long enough that for the full range of the parameter that I'm scanning, that I know that the system will reach steady state. Because the integrator that BioNetGen is using, CVOD, is a stiff integrator, you can give it a ridiculously long simulation time, like 1E12 or something, and it will very efficiently integrate to that point, just as if you had given it an end time of 10. So you don't really need to worry too much. I typically, you could set simulation time to be 10,000, for example. The number of points doesn't really matter. When you do a parameter scan, it's only going to report the value at the end of the simulation. However, it will keep the time courses for every single simulation that you ran. So if you want to look at individual ones, you might want to pick an informative number of time points over that range. But here I'm just going to set 2. You can set different simulation methods, but I'll select ODE for this one. It's an accelerated form of SSA. So okay, and now I can hit run when I filled out all those options. And so I feared. So it said it says here that the simulation does not reach steady state by 10,000. And I'm not sure that I'll have to try it again. All right, now it worked. So I had to set it to 100,000. It can be the steady state criteria could be a little finicky. So
then this is the final.
Oh, I know what it was. I know why that happened actually.
So this prints out the final value or the 10,000th value.
Yeah, yeah. Probably if I want to do SSA, I could have done. I probably need to pick a more reasonable number of time points, and then it would work. Yeah. All right. That was my fault. For it to determine steady state, you have to chop up your interval into a sufficient number of points because it's going to check the last time point versus the time before that.
Oh, so
and before I had only 2 outputs, so it was checking at 10,000 and 5,000. So that wasn't quite. It will not converge except if it shouldn't. It will never, and it will give you the error like it did there. It's not clever in recognizing periodic orbits.
So you can't set it to do the parameter sweep so that it checks at a given time point?
If you wanted to, you can pick any time. You can pick that the simulation time to be any value you want. It's just going to only run it up to that point.
Run it up to the point of convergence, right? Or the point, or the time, or you run it up to the point of the end of your time point. Like it's 2 different.
Right.
With log Volterra, one situation it would work, other one it wouldn't.
Right, right. The steady state flag could be it's useful if what you just like. Sometimes you have a model that you're varying a parameter that may really change the convergence time for that model, and you just want to be absolutely sure that you're not seeing at the end of the range that the model is converging really, really slowly.
Oh, I see. So if you unclick the steady state.
Yeah, I almost never use it, but because I know better than to, like what I'll do is I'll pick an end time of 10 to the 4 And if I'm really worried about it, I'll just pick another time of 10 to the 5, and if they're the same, then it's converged, and I'm not worried about it. But it can be, the steady state can be useful if, because it will cut the simulation off much shorter once it realizes that steady state is reached. So, yeah.
Is there a more high throughput way to do the parameter scans for multiple variables?
Yes, but not, but basically using scripts.
Okay, so it's all accessible via scripting.
Yeah, absolutely. Yeah, you could do that. We actually have an experimental version of a parameter scan script that lets you use different methods of varying parameters.
Sorry, all of this is accessible. Everything that we've done so far is accessible via scripts.
Yeah, you can. So you can run. BioNetGen is just a command line program, so you could very easily write a script that calls BioNetGen and changes. I mean, the easiest way to vary multiple parameters is to make a script that writes the BioNetGen input file, or at least the part of it that defines that sets parameter values, and just have it make a very large input file that changes all the parameters and then run simulations. But not from the interface currently, not from RuleBender currently. You can only do one parameter at a time from RuleBender currently.
What is the tolerance for the steady state criteria? I know that.
It can be. I think it can be customized from the command line version of it, or from the in the BioNetGen file itself. But this interface doesn't let you set the tolerance for that. It's not a heavily, it's not something that we have made bulletproof. Let's just say that.
The simulation method here supersedes the one that we wrote before, because we wrote two different methods for the scan and for the single simulation that we wrote in the code.
Yeah. So you're concerned about, yes. So when we do the parameter scan, that's a really important point. It's ignoring the actions in the file that are down here. It's only dealing with the stuff that's in this begin model and end model part of the. So strictly speaking, in BioNetGen, you can make the model without enclosing it in begin model and end model, and that's fine. But if you try to do a parameter scan, it will give you an error if you don't enclose it in begin model and end model, because it's looking for those to make sure it knows what to do, what to ignore.
It's generally like a safe thing to put.
Yeah, it's a safe thing. So, we wrote the scripting language so that you could, to be as convenient as possible, so you could do everything within one file. So you didn't you didn't have to worry about transferring around multiple files, and because I hate that, and I feel like then the model gets disconnected from the input, and you can't figure out how to reconstruct that. So we set it up so everything goes in one file, but sometimes you want to be able to separate the model itself from what we're doing to the model. So that's what everything that comes after the end model are what we call actions. So, okay, so we looked at the scan, but the RuleBender is lacking one key feature, which is when you do a parameter scan in log scaling, it doesn't automatically log scale the axes for you, so you have to do that manually. So the output of the parameter scan is a lot more sensible looking in log scale than it is in linear. So if you log scale on the x-axis, you could see a very standard isotherm, I guess you call binding isotherm, with a half maximum right at 10 to the minus 9 molar, which is exactly 1 over km here. So if we go back to the file, we see that the k plus 1 over k minus 1 is 10 to the 9, so that's molar. That's 10 to the minus 9. That's 10 to the 9 per molar, which is 1 nanomolar, and that's exactly where the half max is. So these are good things to know. So this looks pretty reasonable. So I said there's another method. So the other method for doing a parameter scan, and this demonstrates just general, that there are more complex actions than the simulate command that we can do in BioNetGen. So if we go back to the web page, there's a parameter scan command. It's basically doing exactly the same thing as what we just did from the interface. That was interesting. If we copy that in, it doesn't quite fit on my restricted window here. It's going to do exactly the same parameter scan as we just did. And the difference is that I run that by, instead of doing the parameter scan, I go back to just run file. So this is going to just run the BioNetGen file with that extra action. Make sure I save it first, run it. Oh, is it going to bark at me?
No, it just gives you a blank screen. It doesn't give you a...
Oh, that's cool.
So you copied and pasted the command that I gave? So who got this to work from the input? Okay. And anybody not get it to work?
I mean, it's been on and off for me.
Oh, yours is going to be a little weird because of the Java thing.
No, but even on this computer.
Oh, on that one. On what's off mean in this case?
Like not working.
Oh, you're using a different model?
I'm using, LV.
Oh, that's the like voltage. So the parameter scan will not, certainly if you click steady state, will not work because it doesn't have a steady state.
I'm not actually, I think I was still talking about zooming. I'm talking about something completely different.
Okay. Did someone get a weird parameter scan result? James, you said it wasn't, log scaling wasn't working. If you log scale the
zooming doesn't work with log scaling.
Oh, yeah, or it's idiosyncratic. It is working for me here.
If you right click and say zoom out.
That's nice. You messed it. I'd completely messed it up. All right, yeah.
I was finding if I want to zoom anywhere near the axes I can't, but if I zoom out then I can in linear scale. And that behavior isn't reachable in log scale.
Yeah, the zooming in log scale, it works, I would say most of the time in my experience, but if you can beat on it and make it break, it's not. I'll say that I think it's easier to use than MATLAB, but that's up to you to decide whether that's true or not. I mean, I find it, for quick and dirty, it's really great. For details, it can be hard. If you haven't picked the range and if you have to zoom into a very small part of the range, it's hard to see. When I do that, I just click onto the numbers and I look at the numbers straight up. I usually find that more informative. But your mileage may vary. We can, by the way, verify that bound 1 and 2 give exactly the same. Like these are numerically identical. The way we define those two observables. But okay. So that is parameter scan and log scaling. Very useful technique to know for when you have parameters in your model that where you really have no idea what they should be. That's a good way to go.
But even then, I mean, you could pick an output and say, this looks like the most reasonable output, but you still don't know if that's the right one to choose.
Sure. There's no...
The best ways to measure it.
Maybe so. Yeah. I mean, I actually, I find, one reason I started to, so we always used to define the parameters up at the top. And when we copy and paste, we have a model. We just take it out of the box. But when I'm building a model, more and more I find I like to build the models where I make up parameters as I go along for the rules. And I do things like set all the first order constants I set to 1, for example. So that's kind of like saying that all of those reactions are happening. The kinetics are happening. There's a single characteristic time scale.
So that's modeling
A little bit, yes. But I'm not saying that yet that that's reality, but that helps me to understand what I've got. And then when that doesn't behave the way that I want it to, then I start to say, well, what timescales do I think are going to be different in the system? So as I'm building up the processes in my model, I'm thinking carefully about what do I expect the kinetics to be for each step. And if I build, and I really recommend when you start building your own model, so when you use the examples, it's fine, copy and paste and play around and comment things out. But when you're building models up for yourselves, build them one step at a time and test everywhere. Test at every single step. Run the model, see if it's doing what you expect. Because if you build up a model that has 10 rules in it before you run it, chances are you've screwed something up along the way. And to debug the 10 rules, there's like so many possible places that things could have gone wrong. It's a much more time-consuming task. If you do it one step at a time, I think you have a more robust, you're much more likely to get to the destination.
And
so
you're building your intuition.
absolutely, and you understand what each part of the model seems to be doing. I highly recommend that. You learn a lot by playing around with things. And it's surprising how often the model will surprise you and not behave the way that you expect it to. And you should, when that happens, you should definitely figure out what's going on before you proceed, or chances are you're going to get garbage out of your model. It's very easy to make models that are just nonsensical. And it's really hard to turn the model that's nonsensical into something that makes sense. Another strategy that can work is taking somebody else's model that you know is supposed to behave a certain way and and making sure that you build it up to behave that way. But even that I still think building it step at a time is a good idea. Okay, that's philosophy. Do we want to do one more example before lunch, or should we stop and?
Can we try making our own model?
Can you try making your own models? You mean you want to diverge from the group? Is that what you're saying? I have one more model that's kind of fun we can play with and do as a group exercise, or do you want to do something on your? Is that what you're saying? You want to do something on your own?
You kind of just play with it and use a model that's not made with rules, that's made as the reaction network or something, and see whether we can build it.
Okay, let's do that. All right. So I have a fun model that is it's like one of the sort of proto models for an autophosphorylating kinase, which could be CAMK2, and it was actually originally framed as a model for CAMK2. So the idea is this: we have yeah. It's really great for teaching. So in this model, I think we have a kinase that we'll call K1, and it can get phosphorylated by a kinase K2. And I'm not advocating this as a correct model of any reality, but it's a very nice prototypical example of a bistable system. So this thing can get phosphorylated through an input. And the phosphorylated kinase itself feeds back and can autophosphorylate another instance of itself. So there's a positive feedback loop here. And then the kinase can also get dephosphorylated. And there's a phosphatase that governs its dephosphorylation. And this phosphatase is could be present in limiting amounts. So we're going to explicitly model the dephosphorylation mechanism as a Michaelis-Menten type, where P binds to K1P and then produces unphosphorylated kinase. So I can give you the constants if you wanted. If you're James and you want to do this model on your own, I can tell you what the right. Where did I put this? I think I remember. So here, for this reaction, we have a characteristic Km and a Kcat. For this reaction, we modeled it as linear, and I just called this S. Let's just call this S instead of K2. And for this reaction, I'm assuming second order kinetics of 0.01 1 over molecules 1 over second. I think I want to say this is 10. And this is 0.4. We may have to revisit this number. So if you can write your own, or we'll do it together. Either way. And if you do it on your own, then you can check with the answer that we get. So what we're going to do is we're going to build a model where we can systematically vary S and look at the the fraction of unphosphorylated and phosphorylated kinase here. And this circuit is really pretty. You know, it's a very general architecture that very often the product of some catalytic reaction itself can catalyze that reaction, and that leads to a switching mechanism. Okay, so let's do it. So, okay. So and it illustrates. I think it can nicely illustrate this principle that we can use the BioNetGen as a calculator to kind of like build a simple architecture, build a simple circuit like that, see how it's going to behave, figure out the parameters under which it's going to behave in a certain way, and then possibly scale that up into a larger model or look at a spatial version of it using a tool like MCell. But if we did the original prototyping in MCell, it would be very slow. The turnaround to debugging the model would be much slower than if we do it with our simple reaction calculator.
How many of CAMK2's reaction sites, or of phosphorylation sites, are you accounting for?
This is not CAMK2.
It has nothing. So we've touched a nerve. A good. I mean, this was originally proposed as a model of CAM kinase 2, but it's a horribly simplified cartoon.
It's not using at least.
It's just saying that there's two states, and it's actually a much better model of something like a kinase and phosphatase, where the kinase, the product of the phosphorylation reaction catalytically activates the kinase in some way. So maybe I can think, if it's a lipid, for example, if the phosphorylated form of the lipid helps to recruit the kinase, then you have exactly this sort of feedback architecture. So it's a good model for that kind of system, very simple biochemistry. CAM kinase 2 is a very, very complex molecule.
It's a dodecamer.
Yeah. Yeah, I shouldn't even say it's not even a molecule. It's but it's a beautiful model because it has this nice architecture. Okay, let's call it.
We're missing a substance to an reaction involving S.
Sorry, what reaction involving? The S reaction. I'll show you how we model. We're going to model S in a very simple way here. So I'm just going to model S as a parameter, and that'll be my only parameter. So we're going to have. Let's just call it K, and we'll have an unphosphorylated and a phosphorylated form of the K1, and we'll have P, which has a binding site. I don't know. I think I called it B. And then you can see my typing is terrible. We could have used the template. You guys are welcome to use it. I'm just trying to be as expansive as possible here. So we're going to have a certain number of phosphatase molecules. I think I set the number to be 10 in my initial model. And of kinase molecules, I want to have more so that the kinase is in excess. I think I set it to be 100. Mm-hmm.
It should be in one single state.
Yeah, thank you. I don't know why I did that. I didn't even think about it. I just wrote it that way. Very good. So if we want to just make it a nice table, we're going to want to read out of this system the phosphorylated kinase, like so. And then the action is going to be here in the rules. Okay, and now here's the fun. So, so the first kind of reaction we know, if we want to model that connection from S, that's just a reaction in which the unphosphorylated kinase gets phosphorylated. And what we're going to do is just make the rate constant be this S. That's the rate of transformation of unphosphorylated to phosphorylated kinase. This could be 0 when there's no input. So, okay. And then to model now the autocatalytic reaction, we just have this. We have an unphosphorylated kinase. Again, actually, just to make the point, I'll copy this reaction. So this is going to be the autophosphorylation reaction. And this reaction requires a phosphorylated version of the kinase. And it's catalytic, so it just, and you know, we could model this more carefully. Mackenzie, are these the right parameters? Oh, that's correct. Is it 0.4? Is that the right one? Yeah. Okay, thank you. We discovered these. I think we took some of the parameters. I can't remember if this one came from Listman or we figured it out something reasonable, but right. And so so these describe the upper part. How does K1 become phosphorylated? There are two mechanisms, and then for the dephosphorylation, we explicitly modeled a phosphatase that's present in limiting amounts. So how do we model that? We're going to model that as a two-step reaction in which the phosphatase binds to the active site at the site of catalysis of the enzyme, and then the transformation occurs. So this is standard sort of Michaelis-Menten mechanism. So here we have the phosphorylated kinase now binds to the unbound phosphatase, and it's going to form a complex. So typically when I do a complexation reaction, I copy the reactants onto the product side and stitch them together, like so. And here I think I think I made the off rate be 1 and the forward constant be. Do you remember, Mackenzie, what?
Actually, I think the 0.4 was.
Here?
Yeah, was there.
I think you're right too. Do you? Oh, this one was 0.01. Okay, and you can see now I've got the kinase. The only interaction in this model is going to be between the kinase and the phosphatase. And now this complex, the K1 phosphorylated, breaks apart, and I can. You have to be cautious when you copy and paste to remember to change the state variables appropriately. But here we go to 0. So this one. You remember what the rate constant for this one is? Okay. 1 seems reasonable. Okay. So we've made that circuit now. And we've written it out in sort of BioNetGen notation. So we explicitly have a phosphorylation site. We explicitly are binding at specific sites between a phosphatase and a kinase. We could put in all kinds of complex regulation involving kinases and phosphatases and other auxiliary molecules as we wanted to, that would start to build up a combinatorially rich system. But for this, it's very simple. And we could have used a simpler notation, but we're just building a scaffold here. So if we want to simulate this model using standard reaction-based approaches, we need to have a generate network command, and then we have to have a simulate command, and we'll use ODE method first. I'm going to guess that this is reasonable. Let's just do 200. I think that's a... What did I do wrong? Oh, I forgot to close brackets. And I forgot to open braces, too. So when you have an editor that tells you where there are errors, you tend to make more errors. That's okay. So unless I've done something horribly wrong, I think we could hit the simulate button and see what happens. So I did build this up. I did build all four reactions at once, contrary to what I told you you should build one at a time. But it's because I know this model. Yeah. And it's probably still a mistake. I probably still made some dumb error along the way.
Pretty easily just comment those out and run them one at a time.
Yep, very easily. So, right. I mean, it wouldn't be very interesting to run this one, for example, one at a time, because I would know what would happen. It would just, I would just linearly produce K1. But that might be useful. If I were showing it to students, I would certainly do it that way. And I would ask them what they expected to see. But OK, let's run this. What do we expect to see? Boom. Not that. Did not expect to see that. I guess I did this too fast. I have the model, but I actually have the model somewhere else. So, so this is a very typical scenario, though, where, OK, so I ran the simulation, and I can see clearly I haven't reached a steady state. So 10 seconds wasn't long enough. And I'm not sure why. Oops.
So I have a quick question.
Yeah.
How do you kill the model, or how do you kill the simulation?
So you ran something that was too long.
It's still going. It's actually stuck at 75%, but it's just not moving.
You can, so in the progress view, you can click on the red button, and it should stop and kill it. Again, I know I cannot make any guarantees with if Java is broken, how the whole interface is going to. It's like there are pieces of it that are broken then, but that should work to kill it, I would hope.
Where is the red button?
In progress view.
Oh. Okay.
Oh, you should be able to. It's an X button. When the thing is running and hanging like that. Oops. Oh, that's exciting. He's done the wrong thing. OK, so let's try 100. So OK, so we see that the characteristic timescale is, more like, it needs about more like 50 to 100 seconds to reach steady state for this system. OK. But we see that with an input of 0.01, only 2% of the molecules are phosphorylated. OK, so let's just for the heck of it, just change the seed species here to be all phosphorylated instead of dephosphorylated. What do you think is going to happen? Any predictions? If you haven't run it, if you run it, or if you did it, don't run it and tell me. Yeah, so so all I'm changing is the initial condition here. So if if there's one stable steady state, I should go back. I should wind up with 2% receptor phosphorylation, or 2% K phosphorylation like before. On the other hand, if there's another stable steady state out there, I might hit it coming from all phosphorylated. So if I run it, I'll find out, hopefully. Oh, I must not have saved it yet. So now I'm running it from the top down. So I'm starting with it all phosphorylated, and I see that, well, indeed, there's another stable steady state. So I just changed the, whether the initially was all phosphorylated or initially all dephosphorylated. There's another stable steady state out there. So this system is bistable, which I just proved. And so which state I wind up in depends on where I started. Okay, and so I might expect that if I change S, the system. So when I change as I change S, I might expect to see at some point a switch where it jumps from this lower stable steady state up to an upper steady state, because that's just from my knowledge of bistable systems, that I know that at some point the lower steady state is going to get unstable, and the system will have to switch. So that's a conjecture that I can now test with a parameter scan, for example. So if I want to do a parameter scan in the simple way using the interface, I have to enclose the model in a begin model, end model, as we discussed. So let me just do that real quick. If you don't do this, the scan will tell you to do it. And then we can go over to parameter scan, and we can vary S. So let's vary S from 0.01 to 0.1, 0.1 to 1.0, and let's just do 100 points just for the heck of it. And I think 10 to the 4 should be enough to find steady state. We'll do log scale, and we hit run again. Oh, that's because it stayed on the upper steady state, and the upper steady state increased gradually with S, but we forgot to change the initial condition. So if I start in a state where the kinase is inactive, then and I do the parameter scan, we'll see a switch. We should see a switch. Yeah, thank you. I put the 0 in the wrong place, eh?
No, you should just have lines for how much each.
Yeah, I mean, you can set the model up in a I set it up with a variable that represents the fraction and change the fraction. But here, oh, it's right at 0.02. Yeah. I feel like I did something different. Okay, so the switching point is right at 0.01. I forgot. I thought it was at 0.1, or I would have done the range a little differently. So if we do this, just shift it over, just for aesthetic purposes. We already know pretty much our answer here. We can see the shift now is in the middle of our range. So we stay in that low steady state going up until we get to S of just about 0.01. It jumps right there, and it stays on. And the signal only very mildly affects the steady state as we increase it over another decade. So there's really a switching that happens. And what we'd find if we scan back from the upper steady state, if we go backwards here, we already kind of did this. If we go down, or if we start from the upper steady state here, the system will stay here. So if we scan this way up and then back down, we'll wind up with all the molecules active. So it's the switching is irreversible.
For the CAMK2 Day2 model? Is there which parameter is the analogous parameter that I can parameter sweep to get the similar?
You're talking about the one that's on the BioNetGen site there? That is we are going to work with that model in the afternoon.
Oh, okay, never mind.
So I'm not sure that there is exactly an analogous input variable. Maybe the calcium, but you'd have to ask Jihan that. You have a new one besides this one. Yeah. Stay tuned.
So what's the end model?
All resemblance of this model to CAM-K2 is completely coincidental. Yeah, exactly. Right. So there is a command in BioNetGen that lets you actually scan up and scan down. And we can do that just for the heck of it as the last thing, and then we'll have lunch. So if I I didn't do the parameter scan. So
yeah, I'm still having trouble with parameter scanning. Whenever I do it, I just get extremely long runs.
My system doesn't like the end model.
This. So you have to put a begin model also
Yeah,
up at the top.
I had to take out.
Yeah.
And that ran fine without either one.
Why does it need it?
The parameter scan, to use the parameter scan from in this.
You have to end the model before the general.
Yeah. You should have to. If it, I think it does. I think it will give you an error if you don't do that. But we can. Let me just find something real quick here. So I'm just going to copy out. So there are two. Here are the command line versions of, or the input file versions of the actions, basically for the parameter scan, and so if you use parameter scan, it looks like this, and we can just replicate what I just showed before with run. Now it's doing it all within one file, and we see there's the switch.
Where's that code? Is that on the RuleBender or BioNetGen?
So the example for the syntax of parameter scan is in the ligand receptor one that I showed before.
Okay.
This Leisman 85 model that I showed you, that one, I made that as prep for this today. But I can post that.
Thank you.
So with the same exact syntax, if instead of calling it parameter scan, I do bifurcate, it will scan up and then scan back. And so then that will enable us to see if there's bistability there. So so let's try that. If we do this, and it will make two lines, one for going up and one for going down. And you can see, so you could see in this this blue curve, this is the result of going back down. So this is backward.
So there's a memory?
So there's a memory here. And the system will switch, and it's irreversible. And that was basically the essence of this Leisman 85 paper, was that with this autocatalytic circuit, you could get a memory of having seen stimulus.
So
And he.
So for 20 years, John, the reason I don't like it is that John got mad at people if their data didn't fit his model.
Right.
He wanted to think that campaigns 2 is member, and it really isn't. So it just, that's why I reacted.
Right. Sure. And I think, it's an example of, there's all these mechanisms out there, and they're really cool. Like, you see this and you're like, wow, that's really neat. And you want it to be reality, but there's no guarantee that it's reality. It's just